Hello viewers, I am back with my next video on carbohydrates. First I would like to thank all my viewers and students also who are watching my videos and giving their precious feedback, which is critically important for me to help improve the content that I am giving you. So please keep watching and please keep giving your feedback. Today I am going to talk about polysaccharides. This is perhaps the last video before I describe the metabolism of carbohydrate. Polysaccharides, as the name suggests, are polymers of monosaccharides. They are also known by alternative names such as glycines or polyosides or polyolosides. These are all alternative names for polysaccharides. They are in fact polymers of monosaccharides and are formed by linking together of monosaccharide units, and whenever a new monosaccharide unit is bonded, it also gives out a water molecule. So in a way they are anhydrides. So we may also define them as polymeric anhydrides of simple sugars. Now talking about their types, we have two distinct types of polysaccharides which are called homopolysaccharides and heteropolysaccharides. Homopolysaccharides are those polysaccharides which are made up of only one type of sugar units. So when they are hydrolyzed, they yield only one type of monosaccharide units. Their examples are starch, glycogen, inulin, cellulose, etc. And the second type is of heteropolysaccharides, which are made up of mostly alternating types of sugar units. So mostly they are made up of two types of sugar units, and their famous examples are like hyaluronic acid, chondritic sulfate, dermaton sulfate, keratosulfate sulfate, etc. One special category that I would like to describe is of mucopolysaccharides. These are polysaccharides characterized by the presence of nitrogenous sugar in their structure, and these nitrogenous structures are, for example, NAG means N-acetyl glucuronic acid, N-acetyl glucosamine, or NAM N-acetyl muramic acid, etc. So they are having those polysaccharides which are having these nitrogenous sugar in their structure. They are called mucopolysaccharides. They can be either homopolysaccharides such as chitin, you see, or they can be heteropolysaccharide such as hyaluronic acid or chondritic sulfate, etc. They are all mucopolysaccharides also. So with this, we'll describe each important category of polysaccharide in detail. Now let me first describe homopolysaccharides. So among homopolysaccharides, as I said earlier, that there are many homopolysaccharides. The first and most common is starch, which is abundantly found in plant sources and acts as a reserve food. So it is one of the most important reserve food of plant. It's a polymer of alpha D glucose, and almost half of the carbohydrates that we consume, they are starch. Inside plant cells, it exists as granules of various forms, which may be spherical or elongated or irregular in shape. Now you see that this starch exists in two main forms. One is called amylose or alpha amylose, to be technically correct, which is straight chain polymer, and another is amylopectin, which is more common, and is a branch polymer of alpha D glucose. First I am going to tell you about the structure of alpha amylose or simply amylose, and I am going to draw its structure. Students, before I do this, I would like to remind you that I have already explained about the pattern of drawing carbohydrate structures, and I will be following Haworth structures. Whenever I will be drawing any structure, I will follow Haworth structure, and I have explained it in pretty good detail in my earlier videos, and I will be using shorthand notation. So for example, in case of amylose, it's a polymer of alpha D glucose units linked through alpha 1,4 glycosidic bonds. So what you are to do, you are to make several alpha D glucose molecules, and you have to link them through alpha 1,4 glycosidic bond. Just see and pay attention to how I am going to draw it. For example, this is one alpha D glucose. As you know that alpha D glucose, we used to draw this like this. This is alpha D glucose, and the numbering also I have told you 1, 2, 3, 4, 5 and 6, and I also explained that it can be drawn like this. In shorthand notation, we'll just draw the side towards which OH is there. So on position 1 we'll just draw this line, on 2 we'll draw this line, on position 3 we'll draw this line, on 4 OH is on this side, so we'll draw this line, and on 6th position this is CH2OH. So whenever we are to draw alpha D glucose, this is alpha D glucose, we'll use this shorthand structure. So once you get familiar with drawing this structure, the pattern of drawing amylose becomes very simple. See how I am going to draw a straight chain. One alpha D glucose, and if you bring second alpha D glucose, for example, here I am going to show this. So here it is OH, here it is OH, and when you link these two, then water is removed and it becomes a, so this position is 1, 2, 3, 4, 1, 2, 3, 4, 5, 6, 5, 6. So this bond is formed between one position of this glucose and fourth position of this glucose. So this is alpha 1,4 glycosidic bond. Now in drawing amylose structure, you are supposed to go on drawing these bonds. See, now you have two glucose molecule. Next we will add one more. Now you have 3, you add one more, you have 4, and so on. So just to indicate that this chain grows like this, I have drawn it like this. So all these are alpha D glucose, all are alpha D glucose units, and all these bonds are 1,4 alpha 1,4 glycosidic. If you go on adding alpha D glucose units like this, the straight chain that you get is amylose. Now you see what is the difference of amylose from amylopectin. The difference of amylopectin from amylose is that it's a branched polymer. Unlike amylose, which had only alpha 1,4 glycosidic linkages, this amylopectin is having two types of bonds. One are alpha 1,4 glycosidic bonds, which form the straight chain, and alpha 1,6 glycosidic bond, which are present at branch points. So I am again going to show you how to draw this structure. See, I start with making straight chain of alpha D glucose. As you see, this is alpha, this is one glucose, then see I connected it with second glucose, then and like this we'll go on adding glucose moieties, and you can see how easy it is to draw the structure. N means we'll go on adding these glucose molecules in a straight chain, and as I written that each chain may have about 24 glucose molecules. So this is the straight chain, but there are branch points also because chains are connected together. So in between this chain at certain point you have to make alpha 1,6 glycosidic. See, these bonds were all these bonds were alpha 1,4 glycosidic linkage. Now at some place wherever there is a branch point, just like as you see here, so wherever there is a branch point you have to make 1,6 linkage. So for example, this is a glucose molecule, this position, so this is 6th position, for example 1 2 3 4 5, and this is 6th position. So if you make a bond with this and connect it to one position of another glucose molecule, so what you get here, this linkage is between 1 2 3 4 5 6. So this linkage is between one position of this glucose and 6th position of this glucose. So this is alpha 1,6 glycosidic bond. So this marks a branch point in the chain, and now we can continue making the straight chain just like this chain. Just like you go on adding, and whenever in this again you can have another branch point, again somewhere else in this you can have another branch point. So these straight chains, each containing 24 glucose units, may be connected through branch points through one alpha 1,6 glycosidic linkages. So when we make these bonds, then we get a highly branched structure, which can be described either as a laminated structure by Haworth in 1930. He explained it like a laminated structure, or it can be explained as a randomly branched structure as explained by Mayer in 1940. So what you see that unlike amylose, which was a straight chain polymer, this amylopectin is a highly branched polymer, and both these exist together in a starch. Now talking about the digestion of starch. So student, you should know that there are certain enzymes which can hydrolyze starch. These enzymes are hydrolyzing enzymes are, I am writing here, hydrolyzing enzymes. So there are alpha amylases and beta amylases. These alpha amylases and beta amylases. Alpha amylases can digest the straight chain at random points, and they yield alpha D glucose units. Beta amylases also digest starch, but they digest it from this end, from non-reducing end. And there is third type of enzyme which are called debranching enzyme. The debranching enzyme cuts an amylopectin chain at branch points or near branch points. The next example of homopolysaccharides is glycogen, which is a stored form of carbohydrate in animals. And you should know that this is very much similar to starch in structure, almost identical to starch except a minor difference. Its structure is also made up of alpha 1,4 and alpha 1,6 glycosidic bonds, just as I described in case of starch. So in the straight chain pretty much looks like that of amylopectin, and this is which is having alpha 1,4 glycosidic bonds, and you see these are branching points. So if I number these molecules 1 2 3 4 5, I think you are familiar with this numbering now. 1 2 3 4 5 6. So this is 6th position, and from this glucose it is 1 position. 1 2 3 4 5 6. So this is 1 position of glucose joined to 6th position of this glucose. So this is alpha 1,6 glycosidic bond. So wherever there is a branch point, there is alpha 1,6 glycosidic bond, and in straight chain everywhere there is alpha 1,4 glycosidic bond. And as you can see that this is the same structure almost that I described for amylopectin. The only difference is that whereas in amylopectin the length of straight chain was longer, it had 24 glucose units. Here the straight chains are shorter comparatively, and contain about 10 to 20 glucose units. So if we represent one glucose molecule, one alpha D glucose molecule with a circle, then you get a pattern like this, which is the structure of glycogen. And you should also know that because this is the storage form of carbohydrates in animals, it is so in our body also, and about 7% of the wet weight of our liver is composed of glycogen. And you will read about the metabolism of glycogen in detail under glycogenesis and glycogenolysis. The next homopolysaccharide that I would like to describe is inulin, which is found in plant sources. This polysaccharide is a polymer of beta D fructose, in which beta D fructose molecules are linked through beta 1,2 glycosidic bonds, and it's pretty easy to draw its structure. See, I am making this. This is beta D fructose. As you already know, this is one beta D fructose molecule. What you have to do, you have just link next beta D fructose, and you know the numbering. This is position 1 2 3 4 5 and 6. So we are taking position 1 of this fructose, and this is OH. So what you do, you remove this water molecule, and then you get this bond, which is beta 1,2 glycosidic bond, and likewise you go on adding fructose molecules in this pattern. Like this, the chain extends, and everywhere This is the molecule called inulin, which is a polymer of beta D fructose. Molecules linked through beta 1,2 glycosidic bonds. The next homopolysaccharide is extremely important, because this polysaccharide is not only the most abundant polysaccharide occurring in nature, but is also the most abundant biomolecule occurring in nature. It is very abundant because it forms the cell wall of plants. I am talking about cellulose. Chemically, it's a constituent of so many things that we see around us. Wood is having around 65% of cellulose, along with lignin, 35% of lignin. Paper is again cellulose, clothes are cellulose. So cellulose is extremely abundant in nature, and it has some very useful properties. It is not digested by many animals, but certain animals have the property to digest cellulose. For example, ruminants such as cow, goat, camel, etc. are capable of digesting cellulose because they have cellulose digesting bacteria in their intestines. Besides, you know that termites also are capable of digesting cellulose. That's why they are capable of digesting wood, and the reason is that they harbor trichonympha, which is a protozoan in their intestine, and trichonympha possesses cellulose, the enzyme that digests cellulose. Talking about the chemical structure of cellulose, unlike amylose, amylopectin or glycogen described earlier, which were the polymers of alpha D glucose, cellulose is a polymer of beta D glucose, and is formed by linking together of beta D glucose molecule through beta 1,4 glycosidic bonds. So to draw the structure of cellulose, you have to make this. First you have to make beta D glucose. For example, this is a beta D glucose, so you have to make OH on the upper side on carbon 1, then carbon 2 like this, 3 here, 4. So OH on carbon 1 is up, and 2, 3, 4. OH on carbon 4 is down. Then again 5, this is 5 position, and then 6 position it is CH2. So this is one glucose molecule. Now if you make another glucose molecule beside this, then what you get? Again this is OH, 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 and here you have OH on this side, and this is CH2. So now you have to make glycosidic bond between these two OH. So once you make this glycosidic bond, then you have to remove a water molecule, and you have to make a bond like this. So this is not like a straight bond, this is like this. So you have to make these glucose molecules, adjacent glucose molecules, and you have to make this bond. This is position 1, 2, 3, 4, 5, 6. So you see that this bond is between position 1 and position 4, and because we are taking beta D glucose, every molecule is beta D glucose, beta D glucose. So this bond is called beta 1,4 glycosidic linkages. Once you know this, the pattern of drawing this structure becomes very simple. You have to make, you have to keep on making glucose molecules like this, and then you have to keep on making bonds like this. CH2OH here, then again so students, you see that this is not a difficult pattern. You have only to make bonds in this fashion. Remember that you have to make beta D glucose, in which OH is on the upper side. So it will go on repeating. As you can see, that this is a straight chain polymer of beta D glucose. So when comparing with AlphaMiros, which was a straight chain polymer of alpha D glucose, this is a polymer of beta D glucose. The next compound in our list of homopolysaccharides is chitin, which like cellulose is very abundant and is in fact the second most abundant biomolecule after cellulose that occurs in nature. It is very abundant because of its occurrence in the exoskeleton of insects, which are quite abundant, as you know. This compound is in fact a mucopolysaccharide because it is made up of nitrogenous sugar. In this case, this is N-acetyl beta D-glucosamine. So it is formed by linking of N-acetyl D-glucosamine units through beta 1,4 linkages, as I am going to show you here. What you have to do, see, this is, if I make this OH on this side, so this becomes beta D glucose. If you make it like this, this is beta D glucose that you are familiar with. If here you add N-acetyl group, NHCOCH3, then it becomes N-acetyl beta D glucosamine. And as you can see, because of the presence of this NH, this nitrogen, it is a nitrogenous sugar. Now, in order to make chitin, what you have to do? You have to make these molecules. For example, OH, NH, and you see here you will have OH, and once you will make a bond between this, then this will look like this. So when you number the carbon atom, this is 1, 2, 3, 4, 5, and this is position 6. Here are 1, 2, 3, 4, 5, 6. So here, students, you see that this is carbon 1 of this neg molecule and this carbon 4 of this molecule, and the bond you get is, because both are beta glucose derivative, so the bond is beta 1,4 glycosidic. Now you go on adding these molecules in this fashion, and the polymer that you will get like this. You have to take care that on position 2 you have to add this group, N-acetyl group, NHCOCH3, and then you have to make this 1,4 bond, beta 1,4 bond. Everywhere it is beta 1,4 glycosidic bond, and the polymer that you get is chitin. Students, the last homopolysaccharide that I am going to describe is pectin, and you know that pectin is a component of cell wall, and it is found in the middle lamella of cell walls. You might have read that the middle lamella of cell walls is made up of calcium and magnesium pectate. So pectin is found in plants and is also abundantly present in basically fruits such as guava or apple, etc. And one important property of pectin is gelation, and because of this it is important in forming fruit jellies. Talking about the structure of pectin, it's a polymer of alpha D galacturonic acid, in which these molecules are joined through alpha 1,4 glycosidic bonds. I am going to show you how to draw this structure. What you have to do, first you have to make galactose molecule, alpha galactose. So when you are making alpha, so OH will be down on this position, then on this position OH will be upon this position, and because this is galactose, so this position 1, 2, 3, 4. So on 4 OH will be up in case of galactose. So this is position 5 and then this is position 6. And students, because this is galacturonic acid, so what happens that in case of galacturonic acid, this CH2OH group changes to COH group. So you have to make this as COH. So now you have this structure of alpha D galacturonic acid. You have to make several molecules like this. Everywhere you have to repeat this structure, and you know that this, because this is OH here and OH here, once you condense these two molecules, then what happens, then water is given out and a glycosidic bond is formed. So this is one position, 2, 3, 4, 5, 6. So this bond is being formed between one position of one galacturonic acid and fourth position of second galacturonic acid. So this is alpha 1 4. Now you will call it, because this is galacto, so you will say galactosidic linkage. Here also you may correct it, galactosidic linkage. So you may go on adding molecules in this pattern, like this, like this, like this, and everywhere you have to make COH at position number 6. So you go on, and what you get is pectin. Now talking about heteropolysaccharides, as I said earlier, that these are made up of alternating sugar units and contain mostly two types of monosaccharide units. Here I am going to describe few important heteropolysaccharides. The first and perhaps the simplest example is specific soluble sugar of Pneumococcus type 3, which is a bacterium. Since it is made up of alternating beta D glucose and beta D glucuronic acid molecules, so its unit is in fact a dimer, and it's easy to draw the structure. I am showing it here. For what you have to do, you have to first draw a beta D glucose molecule. So I am drawing beta D glucose like this. You see that this is beta D glucose, here it is OH, and then you have to draw, maybe I use a different color. This is, okay, this is beta D glucuronic acid. So I have to make it beta, so I'll put this bond upside like this, like this. Here it will be OH, and here, because this is glucuronic acid, so you have to make acid group here. And what you have to do, now this bond has to be beta 1 3, and numbering is like this: 1 2 3 4 5 and 6. Here it is 1 2 3 4 5 6. What you have to do, you have to make one beta glucose, then beta D glucuronic acid, then again beta D glucose, numbering 1 2 3 4 5 6, and then beta D glucuronic acid. And what you have to do now, here you have to make beta 1 3 glycosidic bond. So you have to connect this one position with this 3 position. So how you will do, this O connect to this 3 position. So this bond is beta 1 3, and the next bond you have to make beta 1 4. This glucuronic acid molecule is connected to beta D glucose through 1 4 bond. So here you have to make a beta 1 4. Again you have to make beta 1 3 linkage, and next you have to make, and this pattern goes on repeating. And as you can see that this structure, what is being repeated here is this dimer, means one beta D glucose and one beta D gluconic acid. This structure is being repeated. So this is the structure of soluble sugar of pneumococcus type 3. The second heteropolysaccharide that I am going to describe is hyaluronic acid, which is quite important from physiological point of view. This compound is in fact a mucopolysaccharide because it contains nitrogenous sugar in the form of N-acetyl beta-D-glucosamine, and again it is made up of alternating beta-glucuronic acid and beta-NAC molecules joined through alternating beta-1,3 and beta-1,4 bonds. And how you will make this structure? This is again quite easy, and I am going to demonstrate this here. You see that this is, first you have to make beta-D-glucuronic acid. Because you are making beta, so you on position 1 you have to put this OH above, then down 2, 3, 4, and here because this is glucuronic acid, so you have to make acid group, and next you have to make this N-acetyl beta-D-glucosamine. So to make this molecule, what you have to do, because this is beta, so you have to put OH above. On second position you have to add NHCOCH3. So you can see that this is a nitrogenous sugar. Then you go on making in alternating fashion. Again you have to make glucuronic acid, beta-D-glucuronic acid. This is beta. And now students, you have to pay attention that this bond has to be beta-1,3 bond. So you have to, this is position 1, 2, 3, 4, 5 and 6. So you have to make a bond between position 1 of this sugar and position 3 of this. So this linkage is beta-1,3 from carbon 1 of glucuronic acid to carbon 3 of NAC, and then next bond you have to make 1,4. This is 1, 2, 3, 4, 5, 6. So next bond you have to make between this OH and this OH. So how it will look like? It will look like this, and this structure goes on repeating. Again you have to make 1,3 bond, and again next 1,4 bond. So this is the repeating unit. This bond again will be 1,4 bond. So you see that this is beta-1,3 linkage between beta-D-glucuronic acid and N-acetyl beta-D-glucosamine. And when you go on making these molecules in alternating fashion with alternating beta-1,3 and beta-1,4 linkages, what you get is hyaluronic acid. And talking about the importance of this compound, this you might have studied in the discussion of reproduction, fertilization. You know that I am going to show this. See, this hyaluronic acid forms the cementing material between animal cells. For example, this is human egg, and you see that this egg is surrounded by cells, granulosa cells and theca cells. This is the structure of human egg. And what is ovulated is this thing. This thing is ovulated. So an ovulated egg is having these cells around the egg. This is called granulosa layer, and these cells are cemented together by hyaluronic acid. And you might have read that the sperm, I am making an enlarged view of sperm. This is the sperm. So whenever the sperm has to fertilize the egg, it has to release an enzyme called hyaluronidase. So in this sperm structure, you see that this last part, this thing is called acrosome. So acrosome, this is a structure present at the tip of sperm, and whenever sperm has to fertilize egg, what it does, it releases this enzyme that is called hyaluronidase. What this hyaluronidase does, it digests hyaluronic acid. So when this hyaluronidase digests hyaluronic acid, that cements the cells together. So what happens, that this egg is relieved from these granulosa. These granulosa cells are separated, and the egg becomes exposed to fertilization by sperm, and this sperm penetrates the egg. So you can see that hyaluronic acid is needed for cementing the cells together here and everywhere else. The next heteropolysaccharide is again of very common occurrence. I am talking about chondritic sulfates. So we have few types of chondritic sulfates. Here I am describing chondritic sulfate A, chondritic sulfate C, and chondritic sulfate B, which is also called dermaton sulfate. As the name itself suggests, that chondro stands for cartilage. So these mucopolysaccharides are very abundant in the cartilage tissue. The structure is very much similar to the hyaluronic acid structure that I just described, except some difference. For example, in chondritic sulfate, you see that pattern is almost similar to hyaluronic acid. We are having alternating sugar units and alternating bond patterns, beta 1,3 and beta 1,4. So students, by now I think you must have got enough familiarity with drawing of structures of polysaccharide. So once I give you this formula, shortened formula, you should be able to draw this structure by yourself. Still, just to help you draw it again, I am going to show it here. For example, if you want to draw this structure, so this is beta digluconic acid. So what you have to do, you have to take this beta. Beta means OH above, and this is gluconic acid. So this down, and on this position you have to take COOH, because this is gluconic acid, and here you are going to take N-acetyl D-galactosamine 4-sulfate. So mind it that here it is galactosamine. So what you have to do, you have to draw this. This time you have to make again, means you are again making beta, and this time you are taking galactosamine. So on this position number 4, 1, 2, 3, 4. On 4th position you will have this galactose, so this OH will be above, and this 5, and this is, and this is N-acetyl D-galactosamine. So here you have to make NHCOCH3, and on 4th position you have to make sulfate OSO3. And what you have to do, you have to connect this through 1,3 linkage, means this O connect to 3rd position. So this is beta 1,3 linkage, and in the next molecule again you have to make beta digluconic acid like this, COH here, and this bond you have to make 1,4. So this is position 1, 2, 3, 4, 5, 6. So this time you have to make this bond 1,4 bond. And this structure will get repeated. So I think just like this you can make these two structures also. The only difference is that in this case you have to take N-acetyl D-galactosamine 6 sulfate. So instead of making sulfate on 4th position, you have to take this sulfate group on 6th position. For example, I am going to show it here. The only difference is that here you have to take CH2OSO3H and the rest of the structure is same. It is exactly like this. Only the difference is that here you have taken sulfate on 4th position, here you have taken it on 6th position, because the 1, 2, 3, 4, 5 and 6th position, and the rest of the structure is like this. Similarly, if you are to draw this chondritic sulfate B, then you have to take L-hyaluronic acid and you have to connect it with N-acetyl D-galactosamine. The second difference is that here you have to take alpha L-hyaluronic acid, and because I have described all these sugars before, so you can draw this structure on your own and you should try to practice it yourself. Now see another heteropolysaccharide which is also very common and is found in exoskeleton, which is made up of keratin. So the name itself is suggesting that it's a component of keratin derivative. It is called keratosulfate, and as you see that this is made up of alternating units of beta D galactose and N-acetyl beta D glucosamine 6 sulfate. So again you can easily draw this structure once you know the basics. So here what you have to make beta D galactose. So as you know student that beta OH above, this is 1 position, 2, 3. You know that galactose is similar to glucose except position 4. So on carbon 4 glucose is having OH down, but in galactose there is OH up in upward orientation. So this is 5 and this is 6th position, then CH2OH. So you know this is beta D galactose, and here you have to make N-acetyl beta D glucosamine 6 sulfate. So again you will make again beta orientation, and you have to make N-acetyl. So you know that N-acetyl NHCO CH3 group occupies position 2, then 3, then 4, because this is glucosamine, though OH will be down, then position 5, and this is 6 sulfate. So on 6th position, this is position 6. So on 6th position you have 6 sulfate, CH2OSO3H. So now you have N-acetyl D-glucosamine 6 sulfate. So you have to make 1,4 bond here. So 1 position of this and 4 position of this. So it will look like this beta 1,4 bond. And next bond you have to make galactose, beta galactose like this. This will be up. So this is again beta D galactose, but this bond you have to make beta 1,3. So you see that this position 1, 2, 3, 4, 5 and 6. So this bond you have to make from 1 position of this to 3rd position of this. So this is beta 1,4 bond, and this is beta 1,3 bond, and this pattern repeats. Again the 1,4 bond, 1,3 bond, here again it will be 1,3 bond like this, and this pattern will repeat. So this is the structure of keratin sulfate. Coming to the last example of heteropolysaccharides, I have the very famous molecule which is called heparin. As you know that heparin is an anticoagulant abundantly found in animals. In our body it is found in liver, lung and mast cells. You know that mast cells are tissue phagocytes which contain histamine, heparin and serotonin, and in them the role of heparin is possibly to neutralize histamine. Talking about its structure, you can see that it is made up of alternating units of alpha D gluconic acid 2-sulfate and alpha D glucosamine 2,6-sulfate. So you can see here I have made this. This is alpha D gluconic acid. You can see this alpha means OH is down and on position 6 this is COH group. So this is alpha D gluconic acid and it is sulfated at position 2, and the next is alpha D glucosamine 2 sulfate and 6 sulfate. So it's sulfated on 2 and 6 position. So there are alpha 1,4 glycosidic bonds everywhere. Alpha 1,4, here also alpha 1,4. So this is the structure of heparin, which is an important anticoagulant. Finally, I have to describe some stray heteropolysaccharides which are characterized by presence of more than 2 types of sugars. All the heteropolysaccharides described so far contain 2 types of sugar units, but there are examples where more than 2 types of sugar units may be present. For example, first is vegetable gums. So these gums may have 4 different types of sugars, for example gluconic acid, D-mannose, D-xylose or L-rhamnose. And second example is agar-agar, which is also quite common. It is obtained from marine algae, red marine algae, for example Gelidium or Gracilaria, and it contains D-galactose and L-galactose molecules. Students, this agar-agar is used as a medium in culture, in cell culture, and you may also use it as a medium for separating biomolecules. For example, when you will perform agarose gels, so there you will be using agar-agar. So this is a very important molecule. With this, I am going to end this discussion of polysaccharides. From the next lecture, we will start the metabolism of carbohydrates. So please do keep watching my videos and keep giving your feedback. Thank you.